Welcome. This is Joanne Geer. I am Executive Director for the Westchester Biotech Project. We'll tell you a little bit about ourselves in a moment. In today's program, we are collaborating with our co-chairs for the Institute at the Institute for Life Science Entrepreneurship and welcoming JDRF to talk about a really amazing infrastructure that they've developed for microbiome research. Today's program is on our end on the Westchester Biotech Project, and this is Crossing the Streams with the Microbiome Consortium and our roundtable on translating data science. I want to let everyone know that we are recording the session and the presentations will be available online, but we don't put up the roundtable discussion piece. So when we get to that part, you can feel free to share your thoughts and questions. Today's program will be introduced by a few different folks, and we'll get to that in a moment. But first, I want to share that our co-chairs are Keith Bastian from the Institute for Life Science Entrepreneurship and Eric Vieira, who is at Columbia University. When we first met, he was at CUNY, so he's doing some really amazing work now, and that's me a couple of years ago. We have a really beautiful steering committee. All this is on our website, so I'm not going to take much time here, but we welcome anybody who's listening who'd be interested in joining the committee. Our mission really is to develop communication around enabling technologies and developing interoperability, addressing interoperability issues, and everything you can imagine related to creating, sharing, and analyzing microbiome research data. I'm going to introduce my partner, Michael Welling, who is the co-founder for the Westchester Biotech Project. Would you like to take it away, Michael?
Yes, thank you, Joanne, and good morning, everyone. Thank you for actually afternoon. Thank you for joining us today, which we know will be another sort of stimulating conversation around one of the sort of groundbreaking areas that we have been sort of pursuing. I want to just bring attention to the quote on the screen there, one that we have attributed to a number of folks, but we always overestimate the change that will occur in the next 2 years and underestimate the change that will occur in the next 10. And the amount of impact and change that all of you and your involvement and contributions to our efforts at the Westchester Biotech Project have made have set us in motion down a path to accomplish more than we thought possible in even 10 years. And so we have very ambitious goals and visions and driven by an ambitious mission that we encourage all of you to continue to participate and support in. And speaking of support, none of this would be possible without all of the support from all of our community partners, alliance partners, and of course participants. As you see on this screen, and as these partners continue to grow, it's time consuming to name all of them. But I always like to give a second for people to review the list and encourage you that should you be in need of any insight, resource, guidance, or services from any of these folks, that we certainly encourage reaching out to them, or we are more than happy to make an introduction where appropriate. We have many initiatives underway, and I do not want to take time today to review all of them. But back to the first slide, I want to encourage all of you to not only participate, sign up, participate, speak when you have a chance, but review our website for all of our programming and events, mostly virtual, mostly at no cost, to share ideas and hear what others are doing and help us define how we move forward. So again, I want to thank you all for participating. I want to have a special thanks and appreciation to all of our contributors to today's program, and looking forward to learning some exciting stuff. Thanks, Joanne.
Thank you, Michael. And we just have to say that we actually launched 2 years ago this month. So that's why I like that 2-year quote. We've really been able to reach a broad community of researchers, engineers, and data scientists, and those who love them. And appreciate everyone who's on the call today. I'm going to introduce our speakers in a moment. I just wanted to share that today's program, we are being recorded, and the presentations will be in our YouTube channel within about a week or so, and but not the discussion part that will happen later. It's very helpful if you can mute yourself when you're not speaking. And other than that, we'll be hearing some presentations. Then we will invite everyone on the call, and if I can see your name, I'll say your name. But if I don't call on you, you can certainly send a note in chat that you would like to ask a question. We like to do the presentations first, and then we have a nice long time to invite people on the call to introduce yourself, make comments, questions, and so on. So we really have a roundtable experience. I want to introduce Paul Savage first. He is the co-chair of our roundtable on translational research, on translating data science, roundtable on translating data science. Paul, would you like to say a few words?
Yes, good afternoon, everyone, and thank you so much for joining us. This is an exciting opportunity as I've been volunteering to work with the Biotech Project for the last several months. I myself have been heavily involved with data analytics from the healthcare administrative point of view, and certainly the transitions that are going on in healthcare today are phenomenal. We are very accustomed, perhaps, to having very specific silos of activity. And it's an eye-opening experience for me to hear about microbiome and what's happening in some of the other segments of the healthcare industry. So with that, again, thank you for joining us. And Joanne, back to you.
Thank you, Paul. And next we're going to hear from Tom Richardson, who's the president of the Institute for Life Science Entrepreneurship. Tom?
Yes. Well, thank you very much, Joanne and Paul, for the invitation to participate today. This should be a great program, and we look forward to it. What I was asked to do is to provide just a short overview of the Institute for Life Science Entrepreneurship. We refer to it as ILSE, and just to kind of frame and orient you a little bit to some of the work that we do. And then Bob, who's our head of research, is one of the main presenters today. We'll be talking about some of the depth of his expertise, which is a main thrust of what ILSE does. So if you could go to the next slide, this is really, I only have one major slide here that introduces really what we are and what we do. So the Institute is a nonprofit. We were founded by Kean University, and we're housed at Kean University. We were founded about 5 years ago. And our goal is to integrate all the resources in the region, the New York, New Jersey region, to help develop entrepreneurs, build companies, advance translational research, and build workforce through internship programs. It's right there at the top. And what we try to do is to integrate the science that's coming out of academia, and also that could be spun out of big pharma or the biotech industry in our region, and to help entrepreneurs develop the right type of projects and programs and infrastructure that they need to create companies and develop new economic activity. So we do that across these three circles down below. These are essentially our business units. So in the middle, I'll start in the middle, a business incubator. We have a physical facility at Kean University, which is about 16,000 square feet. About half of it is laboratory space. We have about 8 companies now, 35 employees, about $12 million or so in NIH funding, largely through various SBIRs and so forth. And our incubator activities that we provide are bolstered by a core lab network of companies that help support these small firms with all types of research and product development, essentially the early stage preclinical development work that leads to IND. We also have a large consulting network of over 400 individuals across the globe that literally span the full range of early discovery through clinical development and regulatory affairs, through legal, finance, IP, and everything in between. So we have a big network of people that are available to work with companies based on what the company needs. Now, shifting to the left, our accelerator function We also do a fair amount of work through acceleration. The second bullet is events. You know, things like this program today. We host a variety of webinars and on-site events. Many of you have come to our place in Union, New Jersey. And recently, we just joined CARB-X, which stands for Combating Antibiotic-Resistant Bacteria. This is a public-private partnership funded by BARDA, NIH, the Wellcome Trust, Bill and Melinda Gates Foundation, the US, the UK, and the German governments to the tune of $550 million, focused on developing essentially therapeutics, diagnostics, and vaccines to address the emerging threat of antibiotic resistance. Big Pharma has pulled out of the space. CARB-X was formed to help address it, and they put money directly into product developers based on their science and a lot of great, exciting, innovative work coming on the antibiotic space. We are an accelerator, so we're one of 10 accelerators globally, 4 in the US, 5 in Europe, and 1 in India. And we work with almost 10 companies. I think we're at 9 or 10 companies now, where we're working to help them with their science, their business, their clinical development needs, and really helping them to work through and think through some of the things that they need to develop their products. And we do that in part from our own expertise with folks like Bob, and at the next slide I'll show you some of the folks as well. But we do that through a variety of mechanisms based on what the companies need. Actually, if you can go back. So CARB-X, we just launched this back in February, and we're one of their premier accelerators now, and we'll be working with them at least for the next 3 years. On the right-hand side is our research institute. Bob will talk about some particular area that he's interested in, but we also do a variety of things on the Research Institute front. We have a longstanding collaboration with ATCC. We've created the Center for Translational Microbiology. Many of you are familiar with ATCC. This is the world's largest biorepository for cell lines and bacterial strains and so forth. And we have a 10-year collaboration with them. We're in year 4 right now. We do a variety of projects on antibiotic evaluation, strain development and characterization, microbiome, and bioinformatics work as well. Through our research institute, we also write a lot of grants. We currently are we're PIs or co-PIs or subcontractors on over 8 grants right now from the NIH, NSF, and the New Jersey Economic Development Authority. So we do a lot of work with academics all throughout New Jersey who partner with us for a variety of things. The last bullet there is microgenomics, which is a brand name for our contract research services. Through the work that we do on antibiotics and assay development and so forth, we've got a group within ILSE that performs contract research services, and we have over 20 clients to date. About 8 are active now, and we do all types of MIC testing and bioassay development and so forth, largely in antibiotics and bioinformatics. So these clients are from all over the world, in Israel and Europe and all over the United States. So the next slide shows some of the people involved. So Keith Bastian, who was introduced earlier as one of the steering committee members. I am on the top and to the right there. Bob is on the bottom. And then we've got team members, Ajay, who runs our project management, Steve Lasaic, our head of finance. And then we've got others who are collaborators with us. David Perlin, the executive director of the Center for Discovery and Innovation at Hackensack. He's a former head of PHRI at Rutgers. And his team member below, Vero D'Artois on pharmacology. She is now with the CDI as well. She moved from PHRI. And then Joel Freundlich, who's a medchem person still at PHR and Rutgers. So these are the folks that are working directly with our CARB-X accelerator. So with that just kind of gives you an overview, a brief overview, and I thank you for your attention. Joanne, back to you.
Thank you, Tom. And I have to say that the ILSE facility is absolutely beautiful, and we're in the early stages of developing a program that's probably going to happen either late this year or in 2020 to bring folks together in real time and space.
Oh, that's great. That's cool. You know, well, thank you. The building is nice. We're full. We have no room to grow. We actually won an Innovation Challenge Award from the New Jersey EDA last year. And just yesterday, we had our final deliverable. Our project was to expand our incubator and to create another 20 some odd thousand square feet. So we put together a business plan and marketing prospectus for what that would look like, an internship program with the university. So we're excited by that. And we've got a lot of traction that we're hoping to make good on and expand our footprint here in Union. So that story remains to be written, but we're excited by that.
Oh, that's beautiful. It's a wonderful what you've accomplished in the few short years that you've been in existence. It's really terrific. Keith would be with us today, but he's in China today, so Tom has taken over for him. It's nice to hear your voice. Thank you. So I want to introduce Jessica Dunne. She's Director of Discovery Research at the Juvenile Diabetes Research Foundation. Jessica, I'm going to make you presenter. Here you go.
All right.
So while she's getting ready, we're going to be hearing a couple of presentations, and then we are going to invite folks on the call to join the discussion. If you have a particular question, please put it in the chat box, and we'll make sure that it gets addressed when we get to the second section. There you go, Jessica. Thank you.
You can see me now? You can see my slides?
I can see your slides.
Okay,
Yep.
Great. Want to make sure it was all good. Okay, thanks, Joanne, for the introductions, and thanks for inviting me here today. I'm here to talk about an initiative that we started 3 years ago to help facilitate some of our microbiome work. And so we call it the JDRF-MIBC, or the Microbiome Initiative Bioinformatics Center. So I'll spend just a couple of slides just really orienting you about why we're even thinking about it, and then the last few slides talking about what we're doing in this space. And so as Joanne mentioned, I work for JDRF, which is the world's largest private nonprofit funding type 1 diabetes research. And so probably about 7 or 8 years ago at this point, the microbiome was just starting to get into the forefront of scientific discovery for diseases other than the traditional things that the microbiome had been implicated in, and certainly allergic, autoimmune, immune-mediated diseases, just given the real connection between especially the gut and the immune system. And so one of the reasons we became interested in it is because type 1 diabetes is on the rise. So if you look at this data, which comes from a study called SEARCH, and just really looking at the rising incidence of type 1 diabetes among people in the U.S., and it's, as it says here, it's expected to roughly triple in the next 30 years. And so that cannot be accounted for by genetic drift alone. So it's long been thought that there are environmental factors at play in the development of type 1 diabetes. And in fact, if you look at other autoimmune diseases, autism, allergy, the rise is pretty concordant with what we see in type 1. So really suggesting that there's changes in our environment, and there are actually some studies looking at, underdeveloped third world countries and comparing them to the U.S. and other Western countries, showing that the sort of our modern way of living is affecting potentially our microbiomes and disease outcomes as well. And so we again sort of had a long history in trying to look at environmental factors. And back in about 2011, we started to look at the microbiome, and this is how we established our microbiome consortium. We have a collaborative network, mostly right now, of academic researchers, but also, we've had in the past, but I don't think currently, some industry-based folks as well. And most of the people within the consortium are funded by JDRF, but not all. But the focus is obviously to work on how we can understand how the microbiome is associated with type 1. And the overall goal is really to the overall goal is to develop therapies to get to patients, but where we are sort of now is understanding how the mechanism and identifying targets for therapeutic effect. Okay. And so this was one of the, I think, seminal papers that was published in the type 1 diabetes space. And I'll take a couple minutes just to go over with you just to understand, and then I'll get to the informatics center after of this. So we have been blessed in the type 1 diabetes space with birth cohorts. So the average age of diagnosis for kids with type 1 diabetes is between 7 and 14. And again, we've sort of been blessed with a number of birth cohorts sampling longitudinally from birth, collecting all sorts of clinical samples, including stool samples. And what happens prior to the development of type 1 diabetes is that individuals become what we call autoantibody positive. So they develop autoantibodies, and that's sort of the indication that they are within the next, the average time to diabetes diagnosis then is about 7 years, that they will develop 7 years over the course of their, they will develop type 1 diabetes over the course of their lifetime. And so this is sort of the key point in the development of type 1 is really the development of persistent autoimmunity. And what this basically shows is that individuals, once they become multiple autoantibody positive, meaning they are going to progress to type 1, their microbiome shifts. And this is when they have disease onset. So our thinking is if you could change this shift and really push it back up, these individuals have yet to become and will not and may not become type 1 diabetes. So really giving us some believability that we can affect the microbiome for therapeutic effect in type 1. And so where we are is trying to unravel the role of the microbiome in type 1. We feel that, you know, we've got at least a big stake in the field in terms of understanding how it fits in type 1, but we're always looking for partners and ways to collaborate and work with the broader community in terms of how we can understand what the role of the microbiome in health and disease, and also with respect to development of a healthy microbiome. And one of the things that sort of struck us when we started developing the consortium was how difficult this really is. So if you look at even in our small consortium of about 20 funded investigators, we've got everything from animal models to taxonomy to systems approaches to even the cohorts that we're working with to interventions, human immunology. And we didn't have a way to bring this all together. So we at JDRF were sort of the center and the core of the consortium, but we realized we were publishing and producing a lot of data, and it wasn't very well coordinated in a way that we felt like a lot of these birth cohorts are small, and how could we extend those findings beyond what the individual lab had found. And then the second piece of it was many of the individuals that were working in this space really were hardcore type 1 immunologists with very little computational background, and really everybody was looking for different ways to, you know, help in terms of establishing their analytic pipelines. And so we thought that there was a really big need in terms of establishing that analytic pipeline, as well as bringing the groups together both computationally and in terms of their data. So we, back in 2016, created the Microbiome Informatics Computing Center. And so this is based out of Curtis Hutton-Howard's lab out of Harvard University. And these are some of the key players that have helped develop the platform. And so, as I mentioned, it's first and foremost a computational platform for our data deposition and sharing. And one of the things that I think, actually I'll get to it, I think, on the next slide, that I think is one of the powers of this is that a lot of the publications now require you to push their data to different databases upon publication of or even submission of your publications. And so once you've deposited your data within the consortium, within the Bioinformatics Center, they will automatically push your data to the appropriate publications in a way that's compatible with the publication. So again, sort of one more thing that the investigators themselves don't have to worry with. We do have then a uniform interface for data and metadata and organization, so common data dictionaries across the different studies and things of that. One of the other things that we thought would be really key was to develop this web portal and these computational tools. So the computational tools are available to any of the JDRF investigators that log on to the portal, and they themselves can do the analytics, or they can work with the MIBC team to do combined analytics. And quite often what I've found is that there's probably a third in terms of our investigators, a third of them that feel confident enough or have their own analysts analyst, I guess, in-house to be able to use the platform to just do the analysis. And so they're also saving on their own compute costs, their own data costs and things of that nature at their local institution. A third of them will work with the MIBC to do joint analyses, and then a third will almost solely rely on the MIBC for their analyses, which I think is a great option for people who want to get into this space but lack the resources to be able to do the computational analyses. And we've gone through quite a few iterations in terms of what the web portal looks like to make sure that it is sort of user-friendly. And like I said, the bioinformatics personnel, we've got some full-time people to send some part-time people to help support the members in the projects. And I think the best part about this is that JDRF supports it, so it's free and supported to the investigators that are within our consortium. And so this is sort of just a little bit, about what it does. You can upload your data to, again, share it or submit it for publication. One of the things I failed to mention is that there are some baseline analyses that are run automatically upon data submission, which I think is great. And again, free computing, and the MIBC can support new analyses. So I spoke through much of this. The other great thing I think too is that the MIBC team, they've got such expertise in this space that, post analyses, they'll often work with our investigators to talk about what the next planning in terms of their next experiment, and basically anything that they want to do. And again, I think this has been a wonderful resource. And the whole, one of the other goals of this was to support sharing among different groups. And one of the things I'm not going to get into today, but we do have data sharing rules in terms of, so you're supposed to put your data up upon generation. It's, in lockdown for yourself, I think for a pretty generous amount of time. I think we did 18 months, or upon data publication. And then it gets shared after that within the consortium. So it's a nice way for people to start looking across studies, if there are multiple studies using similar techniques and such. And so quick slide, and I think I've got my next slide also talks about the actual what informatics services they have. So again, sort of common sequencing. They've also built some metabolomics and proteomics type of data platforms as well. Statistics, data visualization, and some of the data visualization stuff that they can do is pretty neat as well. And again, you can do it either via exploratory or for publication. And then this is just, again, a little bit more pictorial view of some of the things that they do in terms of their bioinformatics. And so most of the code is public code, but again, it's sort of centralized into their computes and sort of streamlined already to that effect. So it's again, we think a nice resource for the community. And then this is just the web portal. You can also see that we also have a place for protocols. So the group uploads protocols that are commonly used within the consortium, and there's some additional information as well in terms of the current 10 past team members, data and the users is for people. What's available for the users, and obviously log in if you have a availability to find this. So I think I said about 10 minutes. I went a couple minutes over, so I apologize. But I'm happy to take any questions. I mean, I think the key is we're really excited. It's taken a little bit of time to build up this platform, but really over the past year, it's been really started to been used by the community. Right now, it's closed to the Microbiome Consortium, mostly out of a cost perspective. But happy to talk about any questions you might have, how we can use this resource in a broader space, or any other questions you might have.
Well, thank you, Jessica. This is great. I have some questions which I'm going to save until we get to our roundtable, and beautiful. So, Bob, I'm going to make you the presenter now.
All right, great. Thank you, Joanne, and I'd like to thank everyone for taking the time to listen to what I have to say. As a way of an introduction, I'd let you know I am a microbiologist by training, who has focused on microbial physiology and genomics, working in big pharma for the last 20 odd years. I have worked on mainly pure genomics, as in pure strain analysis, setting up a couple of sequencing facilities and annotation pipelines. So as such, I'm not a microbiome person per se, although I have worked in microbiome analysis as well. So this is supposed to be a roundtable discussion, so I thought this would be a very good topic to really get some discussions going. So, because it is a little bit of controversy around these databases and how to do sequencing and things along those lines. This little statement at the top here is probably good to get some discussion going in its own right. Microbiome research is arguably one of the most irreproducible disciplines of the life science research right now. I'm not saying that there's any error or flaws per se. This is due to so many different steps that are variable, and the fact that it is a self-induced variability in the sense that there is no data standardization. People at all the different academic labs have their own pipelines, they have their own sequencing platforms, they use their own primers or decide to use different variable regions of the primers. Then you end up with the whole process of error correction, filtering, clustering, and then finally you get to the meat of the analysis, which is your classification and your taxonomic identification. So there's innumerable variables in here, which makes taking data from the literature and actually bringing it together from different sources to get some information is very, very difficult. And that's where this standardization platform really needs to be developed somewhere along the line to help things out. So most of what I'm talking about is actual pulled from the literature. This is not my research, I'll state that up front. And I think I have references at the end for people to follow up with it a little bit further. So right now, taxonomically, this is a list of what I would consider the primary databases people are using out there. And Silva, RDP, the NCBI 16S, and Green Genes, these are probably the primary databases, although Easy BioCloud is becoming more and more relevant. This OTUX is one I just actually found, which is a variable region database out there. These are all at different levels of update. Green Genes is probably used, I wouldn't say the most, but it's used very readily, and it hasn't really been updated in quite a few years. Silva is expecting a new update very, very shortly, and the other databases will be updated soon, I would assume, pretty quickly anyway. The problem is that their taxonomies are built from multiple sources, and none of them, I shouldn't say, there's very limited overlap in which sources are being used. So the taxonomies building these databases differ greatly. And the other problem is most of these databases are built from public domain sequence data. And if the annotation of the sequences being deposited is incorrect, that gets perpetuated throughout the whole process. So there are errors that way. This is a paper that recently came out, 2017, comparing the databases and the content. And this is just based on the actual taxonomies themselves. And as you can see, Silva, this gives you the relative content of the databases at the various ranks. And what you see is when you get to NCBI or the Tree of Life, you get a huge number of expanding at the genus level, and it appreciably goes down. But when you look at the actual taxonomies to see how they compare based at these various different rank orders, you see, especially even at the genus, how each database has numerous genus that are unique or are only shared with maybe 2 or 3 other databases. So depending on which database you use, you may end up annotating your genome or your microbiome sample with one genus compared to another. And this is just at the genus. Oftentimes the rank order, as in the whole taxonomy lineage, varies, where this genus may be under a different family within one tree from one database to the next. So it's very difficult to look at the data, and this is the primary output in many regards to what these microbiome researches are delivering. Looking at a little bit more detail, again from the same paper, they compared looking at a strict mapping level from the top down versus bottom up to see how the taxonomies compare between these databases. And even here you see comparing Silva to these other databases, the primary database, there's huge variability as to how this works from a top down or even from a bottom up to see where they fall within the rank order. So all of this just at the taxonomy, and we're not looking at now the actual sequence data, but just how the taxonomies are built within these databases really will give you very different or potentially very different results depending on which database you choose. Here is another paper. This is by Edgar on taxonomy annotation in the guide trees. Here they were looking at actual identical sequences between various databases. And as you can see, without me going into too much detail just for time, looking at just identical sequences, there's huge conflicts in the annotation at genus level, family, order, etc. So here are sequences in 2 different databases that are identical across the whole length, but are actually annotated very differently. And this may be due in part to the process of submitting public domain sequences, which unfortunately these databases have no control. They have to go with the information that is provided on the so-called pure authenticated samples. And there are errors there. A little bit more detail on this, you can see that variations all the way up to the phylum level at 100%. This is from the 16S database. Here we have from the RDP training set, which is supposed to be authoritative, where we're seeing the same sequence being in their database with various annotations at, in this case here, 100% at the genus level, but it's 2 different species. So these are all things that you need to take into account when you're doing the microbiome analysis, and it's probably worthwhile when you're doing an analysis to actually compare your data against 2 or 3 databases. Or if nothing else, it would be nice to have a standardized platform where you can reevaluate your data on a standard platform and other public domain data under the exact same conditions. Now, all these differences I'm talking about are only at the level of the very last step, classifying it, not at all the different processing steps for error correction, chimeric removal, and everything along the way. The other issues you have coming into it is which variable region you're going to use. Each of these will have a huge impact on the analysis, as well as how are you going to actually assign the taxonomy. Are you going to use a nearest neighbor blast approach? Are you going to use maybe the classifier from RDP? These all will also have an impact on your data analysis. Here's another paper that was evaluating 16S databases. Here, the data is, this is all based on the V3, V4 variable region using uCluster for the taxonomy assignments. They were looking at 3, Green Gene, Silva, and EasyBio database. And this is from a known mock community. And they found that using a standard pipeline with EasyBio they got better results in general at the genus and strain level than the other databases. But you can see that none of them were actually able to predict all of the taxonomic assignments in the mock community correctly, and there is huge variation. And then in this case, what we have is a standard process, another mock community, looking at different variable regions. And here we're looking at just the ion torrent versus MySeq. And what you see is a huge discrepancy in how these were called. These were all processed in this paper with the Mather implemented RDB classifier against RDP. And you still see huge variability. So this is the same database, but now how the annotation based on the variable region selected, or even in the case of the exact same variable region, what sequencing platform you're using. So look at that, I'm almost on time. In summary, what has been found is, number one, the taxonomy nomenclature can vary greatly between the databases. So you have to take that into consideration. Variation is up to 17%, maybe plus or minus with green genes and silver. And it looks like maybe a 10% error rate in RDP based on some of the papers that have been published. A lot of these are due to not necessarily the taxonomy, but a lot of the genus level is incorrect just due to the sequence annotation on the public domain deposit. There really is a need for an authoritative data source for some of these. The variable region, of course, is always going to impact the results. And the classifier can also impact the results, not necessarily even by using the same database. So in my opinion, that's where I come up with this point that it is irreproducible data at this stage. And really, we do need a standardization of some sort, similar to, let's say, GenBank and RefSeq, where all the public domain data for the genomes is now being reprocessed in a standard pipeline. So with that, here is a list of the references that I collected most of this data for. There are a lot of other points that need to be brought up. This is a very important one, this 97% identity threshold, because as more and more data is being deposited, we're starting to see database, if you want to call it, collapse, as so many individual strains of a species are starting to show more and more point variation that you really need to be looking at maybe the 99 or maybe even the 100% identity threshold.